Hello.
Hi.
Hi, how are you?
Pretty good.
That's
Did
good.
I miss last Monday? I don't remember.
Yeah, yeah, we didn't have a meeting, so I canceled.
Oh, okay.
Yeah.
Um, yeah. No, I've just been told that I need to produce a 30-page or 30-minute talk PowerPoint by Thursday. So,
Oh, wow.
oh, I didn't, I didn't find the peppercorn I wanted and image it.
Oh, wow, okay.
Anyway, the microscope's actually set up for a peppercorn instead of the lovely, oh, they were canola seed, yeah.
Okay, yeah, yeah, I saw that.
Mm-hmm. Anyway,
Yeah.
that's sort of what happened to me.
Yeah.
Sort of. Well, I was sick there for a bit, so.
Oh, yeah, that's too bad. Yeah. Yeah, I mean, we didn't have a meeting last week. I've been keeping up with the special issue, trying to get things organized for that.
Yeah,
And,
is that okay or?
yeah, I think so. I mean, we're moving along. We just need to start inviting people more, and but I think we've got a good outline for it. I'll probably talk about it in a minute. So,
Okay, okay.
yeah. Hello, Karan.
Hi, Bradley. Hi, Susan.
Hi.
Hi.
Just trying to find my webcam. Just,
Here
How was your, yeah. Any updates from you, or?
yeah. Should I start? I'll just give me 2 minutes.
Okay.
I tried two different things, for the outlines that I'm currently working with. So to generate, outlines for more images, more than just 8 outlines, I'm trying a different method in which, let's say there are 2 outlines of the, like, I'll show the contour. Is my screen
No.
okay?
There
Yeah,
we go. Now you can
is
see
it
it.
visible?
Yeah.
Yeah. So like these are the contours, that I'm using, to generate my 3D model. So what I'm doing is to generate more of these contours. I'm trying to, I'm using something similar to an average function. Like if there's a function 1 and there's a function 2. So to compute, let's say, another contour between 2 contours that are there, something like that, to generate, more information, more points for the 3D cloud, 3D point cloud, that I'll be generating. So it uses a similar transformation formula, like if I want, let's say, 10 contours between 2 particular types. So if I want, let's say, this, these are 2 similar, I think. Like, yeah, let's say if you're transitioning from this contour to this contour, if I want 10 more contours which are kind of like, very similar, like it's transitioning from this to this in 10 steps. It's something like that. So for that, I'm using some more functions to just improve the, to smoothen the model more, instead of just 2 blobs of images put together, right?
Right.
And there are like 2, 3 more things that I'm trying out. One is optical flow. Like there are existing, 3D models that rely on optical flow diagram. So it's like you feed the image to a neural net, and it gives out an optical flow map of the image. Something flow map. Yeah. Uh, yeah, something like this. So the object that is moving, the rate at which it is moving, the motion displays the pixel intensity. So if it's like really brightly colored in a particular way, it's going to, uh, show with the particular pixel intensity. So this is another thing, that I'm trying to try out with this model after I, generate the proper number of, like, to smoothen all these contours that are there.
Yeah.
But, so I'll be moving on to that next. So still going. The thing is, my end semester exams were going on. They're still going on. I think they'll be ending by 16th, so I'll be getting more time. So I was just trying this also plus some exams that just gone over. So, yeah, this is the current state.
So the contours are like the edges of the.
Yeah, these contours make up the 3D point cloud. Like this is actually a NumPy array of points that I'm using further down here to generate my 3D model.
Okay.
So this NumPy array is converted from XY to XYZ coordinates using another transformation formula, and then we get the final output. So the thing is, this thing still needs some more contours. So for that, I'm generating averages of these like 2-3 adjacent contours. So just to smoothen the model that is there. Otherwise, that same like we have two images plastered together, and the from one particular side, it's kind of, it looks as if it's been stitched together. Something like, to avoid that issue altogether, I'll be using this.
All right. Yeah, that looks good.
Yeah.
Hello, Dick.
Good morning.
Morning.
Hi.
So that looks good. So you're just averaging the contours are just basically taking the different images and trying to normalize them.
Yeah, exactly.
Okay.
So I'll be having like more than, like, at least I'm currently thinking of using maybe around 32 contours, like between like 4 contours between each contour that is existing. So let's see how that turns out. I'm still trying to apply the proper normalized function, the average.
Right.
Like how to get from this function to that function in 4 steps. The formula that I'm currently using is basically the circle transformation formula. Like, for every particular degree in a circle, the difference of these 2 points that is there, I am dividing that by 4 and then applying points there. It's like I went through 2-3 explanations of doing something of this sort for XY functions. This was the closest that I got, but because there are these parametric equations, for going around the XY axis. So,
yeah, I see. So yeah, I think that's great. Yeah, thank you for that update. So, I guess Dick had a conversation with Hari Krishna, or he had a meeting, I think yesterday.
Oh, yes, he tried simulated annealing, and he's got something else up his sleeve.
Oh, good. Did the simulated? Yeah.
Yeah, I think he's going to segment the images first and try to align the segments.
Right. Yeah, that sounds. Yeah, let's see if that works. So aligning the segments. So this is different, a little bit different than how Karan is proposing things, but.
Yeah, I put the 2 of them in touch. So.
Oh, okay, yeah.
yeah. He's actually in my college only, Hari Krishna.
Oh, really?
Yeah. In fact, even Menak, if you remember Menak, my youth.
Oh, yeah.
Like, we're all. He's my classmate, Menak.
Okay.
So there's a lot of people in touch.
Yeah. There's some. As I put it, I think your techniques might be complementary. So,
Yeah. All right. I'm just taking some notes here. All right. So, yeah. Thank you, Karan. You can unshare your screen now, I guess.
Yeah.
Okay, there we go.
Okay.
All right. So I just had some things to talk about the special issue. So I've been working on a table of contents. And so this is, I don't know where it is. I don't know if this is the right one. I think it is. So this is kind of a table of contents with the different, I don't know if I made the changes. I just have some names here and some topics. So the idea is we have introduction, type of developmental network. So we have like, just a bunch of different types of developmental network papers that would profile this. And again, these are just like names that I've put in here as proposing some content so we can see what it looks like. Then a couple of special sections. So one is networks and tissue formation. That might be a good topic to zero in on. Another topic might be networks undergoing change. So maybe the dynamics of networks, phase transitions, other types of transitions in tissues. And then a final... What was that?
What does star mean?
Oh, it just means I have to invite people, or that these are people to invite. So this is a note just for myself, yeah.
And no star means what? We already agreed?
I think it's just you, me, and Susan who aren't starred. So this is the sort of way I'm going to keep track of this.
Okay.
And then using networks to resolve dilemmas. That's the last part, which would be kind of like different open questions with networks. So, it's not as focused on networks, but it's maybe how to use networks to resolve dilemmas. And in that category, I put Dick's idea about a call for a modern test of Donald-Williamson's species fusion hypothesis. There are a couple other topics that are potentially in here. And then, I don't know, maybe there's another category like, maybe like a machine learning section or something. I'm not really sure how that might fit into maybe one of these categories, or it might be its own category. So I'm just, I'm trying to put a table of contents together, maybe like in these different areas to give it, to guide, how many people are invited in each category. So there's a balance of different topics. And, we might not get to all of these. It depends on who agrees. But that's the structure.
Okay, looks good. Maybe you should send that to the publisher.
Yeah. I'll send something, an update.
Definitely include machine learning in it as possible. Because everybody was using that particular type of thing to, when I was attending APS physics, a lot of people were using machine learning as a tool.
Right.
Bradley, do you have something, standard approach, regulatory networks?
I don't know if I have anyone in. I don't know what that would fit into. Maybe that would be its own.
That's the current dogma.
Yeah, I know. It's something like regulatory networks. Yeah.
Okay.
Yeah. So then that would be, probably something targeting that. I mean, there could be like a specific paper or, the way some of these special issues usually work is they give like an overview and they say, well, we cover this topic and these papers cover this topic. But yeah, it would be good if we could get a regulatory networks paper. All right.
Maybe you should. Okay, types of development. Okay, that's fine.
Yeah. So yeah, that's ongoing, and yet to really kind of get any responses from anyone. I haven't really gotten too many. I've been trying to work on the invites, and then
Yeah, I heard an interesting lecture by a woman who's simulating the motion of individual cells if you take two balls of cells of the same type and let them fuse.
Okay.
Okay, and she may try doing it with different kinds of cells. She did it with the same kinds.
Yeah.
And it raised some interesting questions, like whether the cells stay put relative to their neighbors or do they diffuse through the whole mass.
Yeah.
Okay.
Yeah.
So that could be seen as a network problem. I'll dig out her name and send it to you.
Okay, that sounds good.
Okay, I don't know if Jonathan Colon is doing work with networks, but I just wanted to point him out again because he was doing some statistical analysis, and he said, oh, definitely this gene is not necessary for development. It was actin that was needed during, like he'd done some sort of analysis, and I only caught part of his talk, but I keep sending him his name.
Yeah, I know. I put it on the master. I have a master list.
So
yeah.
interesting. Like the approach was a deep learning approach. Looking at, like I say, this gene, like Hox gene and actin and tinnitin, and he would say, okay, well, this is what was in play here, not Hox gene. It was, yeah, whatever other molecule that's more physically involved.
Okay.
It was interesting. Like I said, I haven't gone back to it. I'll just tell everybody here again. Somebody wanted me to produce a 30-minute speech by Thursday. So I'm not. No, I just everything else just kind of goes away, and
yeah.
I work on that.
What's the topic of your choosing, or is there a specific topic?
No, I have to do get my thesis topic approved.
Okay.
Oh.
So I get to describe optical coherence, elastography, and the tensegrity project that I'm doing, even though it's not finished, and propose that it's a fantastic project. Yep.
Yeah.
Yeah, well, purpose of that is to generate fear in 20-year-olds.
Yeah.
And just give 60-year-olds just headaches. I'm not afraid of it. I'm just going, I can't do anything else. Nothing else. It's just like, just that until it's done.
Okay.
Yeah, no baking a cake. Forget it.
Oh,
Anyway.
shucks.
Yeah.
Yeah, I was looking for some virtual cake.
Yeah.
Right. Anyways.
Yeah.
Okay.
Well, yeah, good luck with that. I don't know. You probably have stuff to get that you can put together into a talk. I mean, it's...
Yeah. This needs to happen for me.
Yeah.
And, yeah, so, right. Anyway, and then I need to present it to my committee, not just my advisors, and then it'll be okay. Then I can check that off my list, and they'll think I'm making progress.
Yeah. Well, that's good. Well, good luck. So, the other thing I wanted to talk about was that Dick sent me a list of projects that I guess outstanding projects he'd like to recruit people for. And I have some too. So why don't we go through these? Okay, so we have... So this is the list that I have. Well, he sent me a list of like 13 ideas or questions, I guess. Some of these are kind of I think we're addressing now, but some are not kind of been outstanding for a while. So, it's good to revisit these and see where we are with them, and maybe get some ideas of what where we might go with this. So the first one is, are Wolfram patterns closed? Experience with 1D Ising lattices. So we talked about the Ising lattices, I think, in January, and we talked about this, but we've never really gotten on with it. I know that Tom expressed some interest in it. Has Tom contacted you, Dick, about that since?
Not lately, no.
Okay. All right. Then there's
the major problem there is selection, because there are around 800 papers on one-dimensional Ising lattices.
Oh.
Okay.
Yeah.
And we can't simulate them all, not in our lifetime.
Right, that's not not plausible. That's okay. Well, that's interesting. I'll just make a note here.
Yeah, the original Ising lattice by Mr. Ising was one-dimensional.
Right.
And it has a funny history. It was supposed to be a model for magnetism and phase transitions that occurred during magnetism, and it was a total failure. And then somebody made a two-dimensional version, and it worked.
Oh.
So that's how Ising became famous.
Not the person who made the two-dimensional model.
No, I have no idea who that was.
Well, anyways, yeah, if someone wants to work on that, maybe we'll find someone.
So these become we turn a 1D into 2D by adding time instead of space.
Okay, yeah.
Okay. And that's basically, Wolfram patterns are all one-dimensional patterns that are propagated over time. So they generate a two-dimensional pattern.
Okay, yeah.
Give me a second, though. Get a paper handy for the second one.
All right.
Let's see.
So this is the second one. Analysis.
Yeah, why don't you go to the third one?
Okay. Yeah. So this third one we've talked about for a long time, several years actually, and this is the unrolling of snail shells. Can we test if the rolls follow Wolfram patterns? So this is where you might, I think the way we envision this is to take like a 360 picture. You take pictures from all different perspectives, and you stitch it into a flat picture that goes from all the way around the shell. So you get the entire pattern. As if you were to unscroll the shell, it would look like it would be flat, and you could see the pattern. And then you look at that pattern and you say, do these rules follow Wolfram patterns? And these are the cellular automata patterns that we know from the literature, Wolfram's book on a new kind of science, but also the broader literature on cellular automata rules. And so this is something that we've not really been able to, get a really good result on. It requires like someone to take some snail shells, put them on a mount, take pictures of, from different perspectives, stitch them together, and then analyze the resulting pictures. So this is something that...
Coming right up, I'm going to put some of the little shells in the microscope.
Oh, okay. Yeah, I was thinking this might be this might be an interesting thing to do with the microscope. Yeah. So.
Yeah, Bradley, oh, can you hear me?
Yeah.
Yeah. I have a collection of shells mostly cone shells, which I collected deliberately because at some point in the life of the snail, the shell got broken and it had to regenerate the shell, and it had to also regenerate the pattern.
Yeah.
Okay, so we can look at whether or not the regeneration follows anything like Wolfram patterns.
Okay. Yeah, I think that's yeah, I forgot about that part.
Yeah, that's an interesting part. The other question, of course, is, you know, they have pretty patterns which look like some of the Wolfram patterns, but do they really follow that particular rule? Because if they're following a particular rule, it would seem from the pattern you could figure out, well, maybe you could figure out the initial configuration, and then you could figure out whether there's a rule that they're following.
Yeah.
It's sort of that's why Tom was interested. It's sort of the inverse problem of, can you go from patterns to rules?
Yeah.
And the closure question is whether or not Wolfram patterns there are 256 of them, whether they encompass all possible patterns of that type, or do the Ising models produce different kinds of patterns?
Yeah.
Okay, so that's the question of closure.
All right. Okay, that's good. And then you said you had a reference for number.
Yeah, I've got it on the screen.
Okay.
Let me share.
All right, let me unshare the screen.
Start sharing. And,
Yeah.
okay. Can you see a diatom across your screen?
Not yet. Try it again.
Not yet. Let's see. Oh, I've got to select the screen.
Oh, yeah.
Okay, share. There we go. Now,
Yeah, I can see it now.
Here. You see it?
Yeah, I can see it.
Okay, this is an aberrant-like morpha. That's a genus.
Okay.
It's done with transmission electron microscopy. So the dark is the silica and the light is the holes in between the silica. And you can see 3 levels of structure here. What are called the stria. Well, in the middle you see the midline of the raphe, except these are aberrant ones, and they have aberrant raphes like this one. And they're kind of interesting in that the patterns look like they run into each other. And they come off of the raphe and run into each other. There are also little cross stria, which one could possibly pick up. So the image processing problem is first to just segment this into the aberrant raphe, the stria, and maybe the cross stria. Okay, so we know where all of them are, and we can get some statistics on them. And these were made by Ryan Drum about 60 years ago. Okay? And I have about 100 of them. And each one is different. Okay, so this will give you an idea. Let's see, I think I might have another one on the screen. Yeah, here's another one.
Okay.
Here's one where the raphe midline is completely broken up into 2 parts.
Oh, yeah.
Okay. Okay.
Yeah.
So it would be an interesting exercise as to whether one can, well, first of all, do the image processing to get some, try to segment this whole thing into the 3 kinds, and then to see if a simulation can be done which would match any of them. Here's another one.
All right.
Okay? So this one has a full circle on it, and the pattern goes both inside and outside of the circle.
Yeah. Oh, yeah, I see that.
Okay?
Yeah.
Are they all the same type of diatom?
Yeah, they're all the same species.
Does this one looks more rounded because it's got because of
Yeah.
the raphe? It
Well,
seems to change its shape.
It could be that the raphe has an influence on the overall cell shape. Maybe that's why. I suppose that would be one of the questions to ask.
Yeah.
Yeah.
Okay.
Oh,
In this
anyway.
case, but the Rafe is a real mess in this case. But, or the, I'm not sure exactly what they call it. It might be called a Rafe sternum or something like that. I have to look up the jargon.
Yeah.
Well, what causes that? Like the difference?
Nobody knows. Nobody knows.
Okay.
Let's see. Here's a normal one. Okay.
Yeah.
D is a normal one. These are some other species. But, okay.
Yeah.
This shows a normal one caught in the act of growing its silica. Okay.
Right.
There's an earlier one, or just started.
Oh, that just started growing?
Yeah, this just started growing. This has grown almost completely. And
Okay.
this will be normal. Here's another aberrant. Short Rafe, radiating. And this might be a different species. I have to check.
Okay.
Now, another problem which I listed there is this one, G. This is a concentric diatom. And what's weird about it is that some of the stria are in groups, and in each group, they're parallel to one another.
Yeah.
And the question is, what on earth rules could you possibly be using there to make such a structure? It's the only picture I've ever seen of this type.
Yeah.
Okay.
Looks almost like a sand dollar. Yeah.
Yeah, a little bit. Yeah. Yeah. These things, I don't know, they can be up to 0.5 millimeter in diameter.
Wow, okay.
Okay. The centrics you get mostly in the ocean. The pennates, the long, narrow ones, you get mostly in freshwater.
Okay, yeah. Yeah.
Okay.
Yeah.
Okay, so there are two problems in these pictures. Oops. Let's get back to your screen.
Okay.
Okay. Sure. Okay. I guess I can stop sharing now. Okay.
Yeah, okay. All right, let me go back to the list. So that was 2.
That was number 2.
Yeah, okay.
2 and... Oh, and 8.
And 8, okay.
Yeah.
So these are both linked.
Well, they were illustrated in that one paper. But, as I said, I have about 100 micrographs for number 2. I only have that one picture for number 8.
Yeah. 100 micrographs, about that.
About 100. Yeah, they're all actually on film. So I have to scan them all.
Yeah.
And we might be working with Vadim Ankov, who simulates diatom morphogenesis.
Okay.
Okay, and he does it at the molecular level.
Oh, okay. So, yeah, that's so we have 4 of those. Then we have this, well, this is the deconvolution of sparse photon counted for data for computed tomography. So this is, I think this is something we talked about a long time ago.
Maybe. Okay, the problem here is in computed tomography, you want to keep you're sending x-rays through a patient.
Yeah.
And if you do it the way it's currently done, dose is about 16 times of a normal x-ray.
Yeah.
Okay? So trying to reduce the amount of x-rays and still get good quality images. And a number of people are now switching to photon counting detectors, which means that they actually count the number of X-ray photons that come out on the other side of the patient.
Okay.
Now the problem is the numbers of those photons can be low, and they're integers. And all of the methods that people, almost all the methods that people use are for real numbers.
Right.
So the question is, how can you get the best image, and it may involve deconvolution, if the X-rays are photon-counted X-rays.
Okay.
Okay?
All right.
So you're converting to digital.
Yeah. Basically, yes, digital X-rays. You're right. You count every photon.
Yeah.
Okay. 5 fits more into what people have been doing.
Yeah. I think we talked about this a lot, about the movement of basilaria.
Thomas Harbich, from his visual but not quantitative measure, looking at basilaria, he thinks it's smooth. The pennate diatoms we looked at were jerky. So the question is there something very different about basilaria?
Okay. Yeah.
Okay. Now, I think the first measurements were done of the jerkiness of diatoms were done in 1979 at 10 frames a second. Currently, most movies that are online for basilaria, I would guess, would be about 30 frames a second. So I don't think anyone working on this has to take any new movies.
Yeah.
Probably lots of movies of basilaria, and they're probably at 30 frames a second. And the question is just analyzing them for how smooth the movement is.
Right. Yeah. And that's data that we can get. I mean, we have some movies.
Oh, yeah.
Yeah. So that's not.
There are gobs of movies online. So
yeah.
Getting data is trivial.
Yeah. And then this number 6 is tensegrity analysis of cytoskeleton with motor molecules. So this is.
Susan, I don't know if you're looking at this. The question is, what happens? See, most tensegrity structures have fixed connections between the solid parts and the elastic parts. Okay.
Yeah. They've made some active tensegrities.
There are. Oh, yeah, I know that. There's a model for a tensegrity structure that can move, that can wander on rough landscapes, which is the purpose of which was we launched to go to other planets.
Okay, well, I have one that's triangular, like it's 3-strut paper, and it was helping me analyze what I was trying to do. But they made it so changeable.
The question here is if one of the, what do you call it, struts for the hard part, or the struts. Yeah. Okay. If one strut is connected to another by a motor molecule, which can move along one of the struts, what happens to the tensegrity structure?
Yeah, well, there's, I was just looking at a paper about the corners of epithelial cells, and that's where you get a huge clump of actin and and that actually moves. Will move along the cell so you get 2 corners coming together, and the other ones aren't necessarily affected. Did I send you the paper, Bradley? I don't know if I did.
I can't remember. Yeah.
Oh, that's great, because there's also work on the shaped droplets where things will move to the corners and sometimes come right off the structure at the corners.
Oh, wow.
And there's an enormous amount of literature on the difference. Oops, sorry, we got a phone ringing.
That's me.
Okay, hold on a second. Susan? Oh.
Oh.
Oh, she shut her off.
Yeah, I think she's taking a phone call.
Hi, it's okay.
Okay.
It was for Jim.
Okay.
Okay. Just your husband. Who cares?
Yeah. Why he couldn't answer it, I don't know. Anyway, yeah.
Okay. What was I saying?
Shape droplets.
Oh no, there's enormous literature on the difference in proteins that are membrane embedded on the surface of a cell and at places where it has high curvature.
Oh yes, because there's more energy on a curve.
Well, yeah, and then you also get segregation of proteins between the curved parts and the flat parts, etc. So there's a huge literature which probably deserves reviewing.
Yeah.
And make some sense of it all. But it may be involved in what you're talking about. Because if those proteins are anchored to cytoskeleton inside, they would drag it along.
I'll get this paper and I'll send it to everybody.
Okay. If you want, I could send you the papers I found so far on curved membranes.
Oh, okay.
Yeah.
I'm going just for this project. I'm just saying that the tensegrity structure of the cell, most of the mechanical force is through the nodes, the points where the cells meet.
Probably because there's been...
Ingber said that. Ingber actually, Donald Ingber.
Yeah, that may be because there's been enough time for the membrane proteins to move structures to the corners.
Okay, well, there's...
So in other words, it may be involved with membrane curvature.
Oh, okay. Well, they said that was actually in mature epithelial.
If you think about it, the edge of a cell will have curvature in one direction, but not the other. But at the points, at the nodes, it will have curvature in two directions.
Okay.
Okay, so there's a mathematical difference between the two, and that may have to do with the migration of proteins to the corners. I don't know.
Oh, if I could find the paper right now, it would be great.
Hey, you can find it after your talk.
Yeah.
Okay.
Yeah.
It's not going anywhere.
So we have the project Dynamics of Cell Shape After Cell Division in Polygonal Archaea. Was that something that Dick, you were working on with Minoc?
Yeah, I did a calculation for regular polygons and showed that the stable shape of these cells can be a regular polygon. And depending on the parameters, you get a neat peak at a given number of sides of the polygon. The question here is, if you take a cell that's polygonal and divide it in two, then it's obviously an irregular polygon after cell division. So the question is, what happens next? And can we predict what happens next? And can we match up? There's some movies that have been reported on in the literature.
Right. All right. Yeah.
Okay, but I think you can see that if you take any polygon and just draw a line across it, the two resulting polygons are irregular.
Right.
Okay.
All right.
And this is also involved, by the way, with membrane curvature, because the assumption is that I make is that the energy of the whole system is different due to the edges and the corners of the polygons.
Okay.
Okay.
Yeah. Then we have this project, Solubility of Mixed LD Peptides and Membranes.
Yeah, this is a little bit sophisticated because we need somebody who is familiar with calculating the three-dimensional conformation of peptides which are short proteins, and doing it in the hydrophobic environment of a membrane. And the idea here is that at the origin of life, you might have had mixed peptides, and does any what's their solubility of a mixed peptide in a membrane, and does it lead to any selection for the L peptides, which are what all living organisms use nowadays?
Yeah.
Okay, so it's involved with the what's called the chirality problem. Why are peptides and sugars and whatnot chiral?
Right.
Okay. So it's a possible solution, but it would require some molecular dynamics skills.
Yeah. Yeah, it would. Okay. Yeah. Then there's another bioinformatics. Oh, go ahead.
I just put the reference that I was talking about in the chat.
Okay.
And it's something about Synaptopodin.
Oh, yeah. Okay, you did send me that paper. Yeah.
Okay. Yeah, that's, well, I'm going to use it for this write-up.
Okay.
That I'm doing for tensegrity.
Yeah. Okay. Then let me go through these.
I would.
Oh, go ahead.
Sorry, I would like somebody's opinion as to whether a mature epithelial cell does have just its corners supporting its structure, or whether they're the other. I don't know, what would you call them? Other structures are just holding the cell membranes together, or are they part of the structure?
Right.
I don't know.
Okay. Yeah, so then the last four projects are, this Williamson's mosaic or organism hypothesis. This is another bioinformatics project or related project. And then there's, do cracked diatom valves indicate potential pre-stress? That's probably some sort of image processing or making inferences from image processing.
Yeah, B plus mechanics. If you look at scanning electron micrographs of diatom valves, sometimes they have a crack in them. And it's an obvious crack, and the question does that indicate that there was a pre-stress? Pre-stress means it's stressed before you observe it, I guess.
Okay, yeah.
The famous examples are, if you make do you know what tektites are?
Yes.
Okay, tektite, they're the little droplets of molten rock that froze as they got splattered out from a meteorite.
Okay, yeah.
And if you scratch them, they explode.
Oh, okay.
Suggesting that they're under tremendous what's called pre-stress.
Okay. Yeah.
Okay. So I'm wondering if it would be interesting to collect diatom pictures that show cracks and see if we can make any sense of them in terms of possible pre-stress.
Okay. Yeah. Then these last two projects are, can the 24 squares squared be fit on a 70 by 70 board? And what is the speed of randomization of a line? Yeah. Yeah,
these, as I said, these are just for fun.
Oh, okay.
Martin Gardner, who used to work for Scientific American, posed a problem that if you take the sum of the squares of 1 to 24, they're equal to a square, which is 70 by 70.
Okay.
Okay?
Yeah.
And then he can ask the question, okay, if you make a 70 by 70 board, can you put all 24 squares on it? Yeah. And it took decades before somebody finally figured out the answer was no.
Yeah.
Okay, this is a perfectly useless problem.
Yeah.
And I think somebody has proven that this is there are only two cases where the sum of the squares is equal to a square.
Okay.
And that's 1 and and 24.
Yeah.
Okay, so it's not very generalizable. In any case, I raised the question whether the same problem will work if you have periodic boundary conditions on your 70 by 70 square.
All right. Yeah.
Because I mean, I worked on this problem decades ago, and I was able to prove that I never got the full proof, but I was able to prove that certain squares could not be on the edges. But if there are no edges, then maybe they will go.
Yeah.
Okay. So it's a question. I'd say it's a question of writing a program that tries to put squares down and see if it ever succeeds.
Yeah. Yeah, that's maybe something like when it sounds very A-life related, like something that they would try to do, like with.
Yeah, and you might get better and better, or you might want to start all over, or whatever.
Yeah.
Or you might want to run the program for your lifetime.
Right. Well, that's good. Well, thanks for those questions. We'll have to try to recruit people for them. And
okay.
Yeah.
So the last one is kind of another amusing one. Where are you?
Oh, yeah, I can share my screen.
Yeah, it comes from doing laundry.
Okay.
Okay. You know, there are many laundry problems, like what happened to the missing sock.
Oh, okay, yeah.
I've written a paper on that.
Oh, yeah.
Okay. Okay. The question here is, if I wear a week's worth of shirts, they get all mixed up, and when I hang them up, I hang them so that I won't use them for a while. Right. So I put them to the right-hand side of the closet.
Yeah.
And take only the ones on the left-hand side. And this led to an interesting question, at least interesting to me. I can't find any answers to it. And that is, if you have a line of objects and number them, 1 to n, and you take a small block of them from 1 to d or something, a small block of them, and then randomize them and put them at the end, how long does it take before the whole thing is randomized?
Okay.
Okay. The obvious answers are at the boundaries are if you only take one, if you do laundry every day, so to speak, you only take one shirt, you never randomize it. And if you take all the shirts, wait until you've used all the shirts, then the randomization is in one step.
Okay. Yeah.
Okay. So we have boundaries between 1 and infinity.
Yeah.
Okay. And I'm trying to program it, but I'm running into problems with Mathematica. I have to learn how to debug in Mathematica because I'm getting silence from the program instead of debugging statements.
Oh, yeah.
Okay.
Yeah.
Okay, so those are just for fun.
Yeah, yeah. Okay, actually, I do have the paper Susan mentioned in the chat. This is the one, Synaptopodin is required for stress fiber and contractome assembly at the epithelial junction. So I think it's in this list of papers that I had. She sent me, I think it was sent to me. Yeah, this is it here. So why don't we go over this paper? It's sort of a last thing for today. So this is... So let's go through the abstract and then, see what kind of figures that they have and we can have a little discussion about it. So the abstract reads, the apical junction of epithelial cells can generate force to control cell geometry and perform contractile processes while maintaining barrier function and adhesion. Yet the structural basis for force generation at the apical junction is not fully understood. Here we describe two synaptopodin-dependent actomyosin structures that are spatially, temporally, and structurally distinct. The first structure is formed by retrograde flow of synaptopodin initiated at the apical junction, creating sarcomeric stress fibers that lie parallel to the junction and insert into junction complexes of the apical plane. Retrograde flow of synaptopodin is also seen at vinculum decorated basal junctions. So this is this synaptopodin. I guess it's moving around, or it's flowing or moving around. Do you know, Susan, what they're sort of how to visualize this in everyone's mind?
If you go to the very end of the paper, they have some pictures of the junctions. Basically, the cells fit together. They're sort of hexagonal or larger. There they are. Yeah. And at the junctions of the cells, at the corners, you can see where they've pointed out the what is it called? Synaptopodin complexes. Yeah. And when a cell dies in the middle of other cells, it's a confluent tissue, and so it stays together. Like it does there isn't a hole in the tissue. Right. And this is kind of how it closes the gap.
Okay.
Here. I'm looking at D.
This section, yeah.
Yeah. D and E. You can
these two.
see where the yellow comes together.
Okay, yeah.
So that's the cell. This cell in the middle. It looks like a regular cell that has sort of square corners, and then it becomes more rounded, and then it kind of disappears. Yeah. And the yellow.
Yeah, Bradley, there's a network that's changing.
Yeah, I can see.
There you go.
Yeah, it's a dynamic network. Yeah.
And these pictures all kind of show that. So that's part of what I was modeling with my CompSol multiphysics. Is I was showing a grouping of cells, just a plain six-sided cells that are all the same, which is not true to nature, but that's what I was going to use. And then I was going to show losing a cell in the middle of the confluent tissue and just show how the other cells fill in the gap, and then see if the elasticity and viscosity were kind of the same or different in that configuration.
Okay. Yeah.
Because they say that that's a rosette, and rosettes cause stiffening of the tissue. So if they kind of stay permanently, for instance, then they're going to cause a stiff part of the tissue, or they might change the tissue at a very distant cell. APS Physics had some lectures on that. I wish you could have gone to it.
Yeah.
It was very good.
Yeah.
And it was virtual, so.
Right. So yeah, these are some nice pictures. Yeah. And this is a picture, D, of sort of this. They're doing some cherry staining here for the membranes, I guess. They're showing.
Yeah. Oh, and the other thing is in tensegrity, if you put struts together and attach them to each other, it becomes more difficult to analyze and more unstable, or more unpredictable. And I think that this synaptopodin complex is another tensegrity structure, and so these cells are actually joined together with either elastics or tensegrity structures, and they don't form a type 2 or type N tensegrity because they have intermediaries.
Yeah.
That I don't know if you catch what I mean.
Yeah, I know what you mean.
Okay.
this is nice. So if we go back up to the abstract, their findings, yeah. So that's what they mean by this first part. I just wanted to give people, give like a visual understanding of it. So the contractions of apical stress fibers is associated with clustering of membrane complexes via side-on synaptopodin linkers. So that's what we were seeing. Whereas contractions of stress fibers inserted at the apical junction via head-on synaptopodin linkers results in junction shortening. So there are changes in the geometry due to the synaptopodin. Upon junction maturation, apical stress fibers are disassembled. In mature epithelial monolayer or motorized contractomere complex, capable of walking the junction, is formed at junction vertices. Contractomere motility results in changes in junctional length, altering the overall shape of the cell and packing geometry. So it's this in this monolayer that they're observing it. And so the cells are packed in there. And so you have, I guess, a confluent monolayer, and the cells are changing shape, and so they're responding to local forces, but then also they have to conform to forces that result from that packing. We propose a model of epithelial homeostasis that utilizes contractomere motility to preserve the permeability barrier during intracellular movement. So this is about movement and, yeah.
Bradley, there's one other phenomenon which this might apply to, which they seem to have ignored, and that is wound healing.
Is what?
Wound healing.
Oh, wound healing. Yes, that's right.
Yeah.
Yeah, this is where cells will have to migrate into a wound area.
I remember again a few decades ago hearing a lecture on wounds that were deliberately placed in the ears of rabbits.
Okay.
And they acted counterintuitively, because if you did the calculation on stress in a sheet and made a hole in the sheet, the hole should get bigger, but instead it gets smaller. Okay.
Yeah.
This might explain it.
Yeah. Yeah, it might.
So get yourself some rabbits.
Yeah. So the paper kind of talks about how this sort of motivates the problem and then goes through some of the results. So we saw some of the results already. So very interesting paper, Susan. Thank you for that.
Yeah, Susan, you might want to try that.
Maybe I'll try a fly imaginal disc or something.
No, not experimentally. Try it with your computing.
Oh, I see.
Right.
You take an epithelial sheet, punch a hole in it. That should be trivial, and then see if it contracts.
Oh, okay. Well, I'm working on a grouping of cells, so I could try it. But I'd have to probably put the motor molecules on the vertices. So
I know you'd have to simulate what this paper's about.
Yeah.
Okay, but it would actually be very interesting to have a fundamental understanding of wound healing.
Yeah.
Yeah. Well, that's one of the examples I'm using in my presentation. Like, why do you want to study tissue? Well, you can study tissue because it's developmental, or you're looking at cancer, but really it's just normal wound healing and upkeep of homeostasis.
Yeah,
so,
but if you look at a hole made in the sheet of just elastic material, you'll see that the hole gets bigger, not smaller.
Yeah. Well, cells are alive.
Well, they may not. That may not be the important part. Could be these stress fibers that are important.
Okay. Yeah. I can do that. I can just simply take the middle out and see what it does.
Yeah, that might make another paper for the cells issue.
Yeah.
and there's a difference between short cells like skin cells and tall columnar cells. in their mechanical reaction to things. I have a paper that I found on it.
Oh, okay.
So I'm looking for that.
Well, look up papers on wound healing. I think you'll find hundreds of them.
Yeah.
Yeah. Okay. Yep. Good idea.
Yeah.
Okay.
Right.
Yes.
Sounds good. Okay, well, that's all for today. Thanks for attending. Talk to you next week, or we can send things to each other offline, I guess, too. Okay. Hello. Now I'm going to address a question that we had in our Slack from our community. Jesse Parent had a question about the evolution of sensory organs. So I'm going to answer his question. And so here's his question. It's from the Slack. He just asked a general question to me and others. Are there any good resources in how sensory organs have developed over time / evolutionarily? Specifically interested in this with respect to aspects of analog and digital mapping. So this is a question that we've been talking about in the Orthogonal Research and Education Lab. It's sort of a sub-theme that we've kind of hit upon. And so I don't know if I have any resources specific to that, although I can tell you a little bit about it. So my answer to him was, this is quite hard to approach in a comprehensive way because there's so much diversity. The best way to do this is to pick a specific group of organisms and a specific sensory system. As for the analog / discrete distinction, discrete being another word for digital, there actually are different modes of neurotransmission and intracellular communication that can be understood using that lens. I'm going to put together some resources on this. So there's, I think we've talked about the different modes of neurotransmission in this group. You have all sorts of different modes. You have neuropeptides, you have different types of neurotransmitter, and then you have electrical or gap junction connections, and they all have different modes of transmission through the nervous system. So why don't we start with this, let's see if we can start with this paper. This is a review by T. Ryan Gregory, who does a lot of stuff on genomes. And he wrote a paper back in 2008. This is Evolution, Education, and Outreach. So this is a nice sort of introductory article, education oriented, and talks about the evolution of complex organs. So this is just a very broad overview of complex organs and how they evolve. So this is part of the question that's being answered here. The abstract reads, the origin of complex biological structures has long been a subject of interest and debate. And this goes back 2 centuries. And so, you know, when people were trying to explain the origins of different things in the tree of life, and they had to kind of hypothesize, they ended up hypothesizing on common ancestry, and then, you know, people had to take, you know, anatomical evidence and compare it. And then when we got genomic evidence, it became a little bit easier to find homologies and things like that, deep homologies, because you're looking sometimes across multiple taxa and multiple clades. So 150 years of scientific investigation has yielded a conceptual framework, abundant data, and a range of analytical tools capable of addressing this question. This article reviews the various direct and indirect evolutionary processes that contribute to the origins of complex organs. So they use the evolution of eyes in this as a case study and addresses a lot of the common misconceptions about complex organ evolution. So this kind of goes back to William Paley and Charles Darwin, where they were describing that, laying out the foundations of natural selection. Still, while few non-specialists have trouble acknowledging small-scale evolutionary processes, such as the evolution of antibiotic resistance within populations of bacteria, they often remain uncertain as to how similar mechanisms could account for complex structures such as eyes and wings. And so there's a fair amount of literature on this. The eyes, of course, being part of the brain, the wings being another example of this sort of evolutionary diversification. So this kind of goes over the evolution of organs as complex systems. So this paper is not about complexity per se, but about the evolution of complex organs, biological structures with several intricately interacting parts that function in a sophisticated manner. So the eye is actually very famous. William Paley talked about the eye being this sort of perfect product of design, and then of course Charles Darwin knocked down that idea, and then now we have good models of the different types of eyes you see in nature and how they're all related to one another. So, direct adaptation by natural selection. So Darwin famously noted that if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. And of course, this is something that's been, natural selection has been shown to be the sort of the driving force behind the diversification of eyes. So it should be understood that the theory in question is not the notion that species are related by descent. Rather, it is the proposal that the mechanism responsible for evolution is natural selection acting gradually on minor heritable differences. So this is something that we have to remember, is that it's not so much that there's this, it's just that they're related by descent. It's that natural selection is actively acting in every lineage to modify these things. This is why you have such a diversity of eyes, because you have a diversity of conditions under which the organisms exist. There are a diversity of historical contingencies that lock certain phenotypes in over others, and then you observe this diversity in the natural world, and you observe sort of the endpoints of that process. So this is something that kind of goes over direct and parallel evolution. So this is just describing the nature of change, and then let's see if they have anything. Okay, here's an example here of how gene duplication acts as an underlying mechanism for this diversification. You get gene duplication, and then you get the divergence of different clades or lineages, and then they have, so on this lineage here you have a gene duplication here, and then two here, and then something in the tetrapods clade that's specific to that clade. You have common ancestors where you get different changes, evolutionary changes, and they're marked on the tree. And so you can see how this happens throughout evolution. This figure B, this is from another paper here, where they talk about how there's a progression of mutations that occurs on this tree, and it results in different types of phenotype. But it's not just gradual change, it's that these mutations can change the way of the complex trait functions. So this is a complex trait, this ancestral corticoid receptor. And so you can introduce a number of mutations at different sites, and you can get different outcomes. For example, this S106P mutation goes from the ancestral corticoid receptor to something that only binds DOC, which is this thing here. Then the L111Q mutation can be added to that, and it results in a more general receptor. So DOC and cortisol can both bind to this receptor. Now what's interesting is that you can go down another path, and you can find that you can actually have a mutation that takes this ancestral corticoid receptor and turns it into sort of a null receptor. Nothing binds to the receptor, so it doesn't really have a function. But it will maybe stick around, and you might encounter another mutation, this S106P mutation. When that mutation is added to this genotype here, you end up with the same genotype or the same phenotype that you would have through this pathway. So you can see that there's this convergent evolution with, well, actually you can have the mutations in a little bit different order, and they result in the same phenotype, but you have this situation where it becomes a specialized thing and then a generalized thing, and then something that's null and then a generalized thing. So you have these different pathways of evolution that can be observed. And so this is a hormone receptor pair example, but this also works for the eye. Then there's this idea of exaptation, which is where you have sort of things that are used for other things. So you kind of see an example of this back here, where you have this receptor that is used as a specialized receptor. There's another mutation, then there's this generalized form here. So this is, again, something that is an exaptation. You get exaptations oftentimes with organs that can be used for other things. So the evolution of wings provides one of the classic examples of exaptation and secondary adaptation. As has been pointed out many times, a rudimentary version of a wing could not be useful in flight because it would be unable to generate sufficient lift. Only when the wing reached a sufficient size and strength could it be useful for enabling powered flight, meaning that natural selection could not favor variance within a population on the basis of flight ability alone during the early stages of wing evolution. So, to get this initial trait, you wouldn't necessarily have it serving the function of flight, but it would serve a different purpose. So it had a primary adaptation. So bird feathers, for example, probably originated for thermal regulation. And so, the rudimentary wings may have been useful for other things. And then eventually those things combined and formed something that was suitable for flight. The same thing with bats. In bats, early skin flaps probably would have been functional for gliding, but not in powered flight. In insects, it has been hypothesized that early wings were used for skimming across the surface of water. And then you add on other adaptations or other mutations to the genotype, and you end up with this complex phenotype that's capable of doing something. And then we're looking at this from the modern day. So we're seeing the bird fly, and we think, well, the wings were directly selected on to adapt for flight, and that's actually probably not the case. It's probably that there were steps, intermediate steps, that led to other, that you could get one function and then a secondary function, and so on and so forth. It's worth noting that in certain lineages or in certain clades, you have changes that occur, and then you can't, say for example, go back to a period where, I guess you could go back to a period where there are no wings, but you're kind of locked into like a certain evolutionary pathway. So you can't, for example, take your wings and evolve 1,000 legs. That would have to be something that would take multiple steps and a lot of time. So this is something worth keeping in mind. And so this is, again, this kind of goes through exaptation, secondary adaptation. There's also scaffolding. So many organs, having been built up in overall complexity by direct adaptation, exaptation, and collage, and further specialized through secondary adaptation, exhibit a level of integration to the point that their components are interdependent on one another. So this is an example of scaffolding. You're just building one trait onto other traits. And again, this is historical contingency in action, where you're building upon different traits, and you're building upon traits, maybe on 2 or 3 layers deep, where you have, complex traits don't just emerge de novo, they emerge from precursor organs or precursors. And so this is something of like how the eye has evolved. You had a very simple light sensing organ, and then it evolved into other types of eyes, insect eyes and mammalian eyes and things like that. So, this is an example, a very abstract example of exaptation in the scaffolding. I know Jesse's interested in scaffolding in the context of cognitive science and the context of cognition, and it's a similar thing. I don't know if you can make that direct connection, but maybe it's worth talking about. And so, actually, he gets into trade-offs, constraints, and historical contingency here, and he calls it non-adaptation. And non-adaptation just simply means that this is not an adaptation. This is actually the sort of options that you're locked into going forward in evolution. So they serve as constraints. Again, you have 4 limbs. Maybe that locks you into wings or 4 limbs. You can't necessarily evolve like 1,000 limbs coming out of that single set of 2 limbs. I'm just giving a silly example, but that's sort of the idea. And sometimes people call that the hopeful monster, and that's where you have these gigantic evolutionary changes. It's usually in the context of development. That's considered to be very unlikely, and the idea was to contrast that with sort of a gradualism that is built. One thing is built upon another. So now we get into these light sensing systems. So this kind of shows examples of simple light sensitive system is shown in A. This is the ocellus. This is found in the larva of the box jellyfish. So the light sensitive cell is not connected to a nervous system of any kind, but instead includes a cilium, which can be stimulated to move the larva in response to light. So this is a very simple light sensing organ. B is this here in the same box jellyfish species where they have complex upper and lower eyes with retinas. And so this is where they build this is a complex eye as opposed to these light sensing cells. And this has a lot more structure to it. And so this is at the end of a sensory club called the rhopalium, which goes to the central nervous system. And then C is this simple eye spot found in the larva of the trematode flatworm, which consists of one pigment cell and one photoreceptor cell. So this is the receptor cell, this is the pigment cell. This kind of looks like something you might find in a more complex eye, but it's definitely, that relationship is already there in this flatworm, or this, I shouldn't say already there, but it's you can see a version of this that's much simpler than what you might see in other eyes where you have receptor and pigment cells. And then D, a slightly more complex visual organ involving a simple pigment cell, but multiple receptor cells found in the Turbellian flatworm. In this species, the pigment cell is cup-shaped, such that information about the direction of light can be obtained by comparing input from the different receptors. So this is an integration organ. And so you have these receptor cells here that come into this pigment cell, and they have different sources. So this is kind of like a complex nervous system where you have multiple synapses on a neuron. And so you see this. And so this is how this works by integrating light sources, comparing input from different receptors. Landon Nilsson actually is a good resource for eye evolution. Nilsson, 2005, it's the same author. These papers are actually pretty good if you want to find out more about the evolution of eyes. This is another example, chambered eyes versus compound eyes. So this is where you get this, these are in different clades. So chambered eyes and compound eyes are different sort of solutions to vision in different clades. And so you see this, so this is, I think this is a pit eye, and this is found in nautilus as well as many flatworms and annelids. It's a basic compound eye, is B here. And so you can see that there are different types of eye that evolve, and they evolve in basic, in different ways. You can have different families of this type of eye, and they have different ways of functioning. And so you can see this diversity in action here. And so this shows an example distribution of eye types across major taxa. So then we're putting these eyes on a phylogeny. We're going from Cnidaria as sort of this outgroup or this comparator group, and then we're looking in the in-group, we're looking as far afield as vertebrates, insects, crustaceans, mollusks, annelids. And then we have all of these different types of eyes. And you can see there in vertebrates you have a diversity of eyes, but it's nowhere near the diversity that, say, you have in crustaceans or insects. And so you can imagine that this is all, sort of have their own sort of historical contingencies and have their own different versions of secondary and tertiary adaptation. And so you're able to see a very large diversity of complex eyes. Okay, so the second paper I want to talk about is this paper, Origin and Evolution of Cell Types. So this is just kind of highlighting a paper that came out in 2016. And they talk about this is about cell types and how they're the basic building blocks of multicellular organisms and are extensively diversified in animals. So with all of these complex phenotypes, they get built with complex cell types, and we saw examples of that in the eye, where you have different cell types that are you have basic cell types, but then you have specialized cell types. And it's kind of hard to know, people have tried sort of categorizing cell types because you can have a lot of different receptor cell types that are highly specialized across species, especially, but also in species where there's they specialize for a certain function, like species that have different colored cones. And so, those cells have to be specialized. They're not necessarily different cell types, but those cells are specialized. And then in evolution, they may actually become different cell types. People have tried using phenotypes, but phenotypes are really insufficient for understanding cell types. You really need to have a lot of molecular profiling, which is done. And even then, it's kind of hard to make the distinction. And so as a result, the number of cell type, that people have made estimates of the number of cell types, and that ranges widely. So here they talk about some of these cell types. Despite recent advances in characterizing cell types, classification schemes remain ambiguous. We propose an evolutionary definition of a cell type that allows cell types to be delineated and compared within and between species. Key to cell type identity are evolutionary changes in the core regulatory complex of transcription factors. So this is something that you see in many cell types where they have this core of transcription factors that are upregulated or downregulated, and that determines the fate of the cell, whether it changes cell type or not. And so in looking at those transcription factors, you can tell, maybe their expression, maybe their genotype, to see, if it's a different type of cell. This makes emergent sister cell types distinct, enables their independent evolution, and regulates cell type specific traits termed apomeres. And so you can see these in, people look for these different differences, and they use phylogenetics to sort of sort this out. We discuss the distinction between developmental and evolutionary lineages. So we talk about developmental lineages in the group. These are cell lineages. But they also exist as evolutionary lineages because they may exist between species, and so they have to distinguish this. And so there are any pictures in this paper. Okay, this is an example of homology, which is common ancestry for a specific clade. So this group of taxa are related and they have common ancestor. The change occurs here, and it's called a homology because they all share this, at least the ancestral form of this trait. Convergence is where you evolve the same type of function in two different lineages. So this might be bats and birds. And then concerted evolution is where it's across all members of the same clade, and maybe they're all sort of co-evolving, or they're all evolving a similar trait. And so, instead of diverging, like you might see divergence in a number of different types of wing, you might see concerted evolution, and they're all kind of in that same clade. The traits are a little bit different, but they're all evolving in the same direction, perhaps. So they kind of go through different cell types. This is an example of regulatory signature of a cell type identity. So they show this example of these transcription factor clusters. And so you see this actually in cellular reprogramming. This is important. They've identified 4 or 5 different transcription factors that, when introduced into the cell, can change the fate of a cell. You see this in development with other transcription factors. Different developmental changes can be triggered. And so it is with cell type, as cell types emerge in development, and we have these changes in transcription factor regulation, and this results in a change in cell type. And so this is just the identity of a cell. So identity of a cell is just what it identifies as. So, different functions can be fluid, and different cell types can be fluid. So, it depends on the developmental system. If you have what they call regulative form of development, which is something like mammals, then you can see this kind of, signaling will sort of influence the cell to have a certain fate or to have another fate. But in other species, in mosaic development, you have cells that have a fixed identity throughout their life history, and so that's something to keep in mind. This is how they look at like cells in mammalian species and other species with this sort of regulative development. And so they want to point out that the model of cell type identity discussed here has important implications for our understanding of cell type homology. Our model implicates the independent control of gene expression, which we link to cell type identity, may be mechanistically dissociated from cell phenotype. So the phenotype may not reflect what the cell is doing in terms of its function. And it is hard, in fact, to identify phenotype exactly, because sometimes it's very cryptic, and it's hard to understand what the cells are doing. Sometimes they're in a transitional state. So, for example, visceral muscles and Drosophila melanogaster also express these transcription factors, yet have a striated phenotype, suggesting that the visceral myocytes co-opted a striated module in an ancestral species. And so this is an example of some of these evolutionary changes and how these might occur. And so that's about cell type. And this one is now more about the receptors themselves. So this is the evolution and development of vertebrate lateral line electroreceptors. And this is a type of sensation called electroreception. And this is actually in basal to vertebrates. In other words, it's at that common ancestry of all vertebrates. And it's been lost in some vertebrates. It's been enhanced in other vertebrates. So we have this, so you see this a lot in teleosts, which are bony fish. You see a lot of independent evolution of this. And so this is, this kind of talks about the sensory system and the diversity that you can observe in this system. And so, there's a lot of opportunity to look at this as a model system. So, this is across organisms. This is what they call a model sensory system. And so this kind of is a review of electroreception and, some of the, so it was first discovered in weakly electric fish, which are freshwater fish in the tropics. And there are a lot of different species. And then they found this in other types of living vertebrates. Different systems, though. The systems have diversified. And so this kind of goes through a lot of this, and it doesn't really talk about the distinction between discrete and continuous, but some of the waveforms that are generated by these type of systems can be very diverse, and the organism has to be able to interpret those waveforms, much as they might interpret electrical activity in the brain or chemical signaling. So this is something that this is a paper that, if you're interested in going over, it's a very lengthy review article. And so that's electroreception, or electrosensation. This is evolution of sensory structures in basal metazoa. So this is, again, in the sort of the trunk of the tree of animals. So metazoans are animals. Basal is that sort of the common ancestor of animals. And they're focusing here on Cnidaria. So Cnidaria have been traditionally viewed as the most basal animals with complex organ-like multicellular structures dedicated to sensory perception. So these are sort of the, there was an early divergence from that common ancestor, Cnidaria, and they have these, they retain these sort of ancestral forms, or what they call basal forms, because ancestral implies that it's the one ancestor and yet it's living today. Sponges have a surprising range of the genes required for sensory and neural functions in bilateria. Sponges, express things like different types of synaptic proteins that we see today in more complex nervous systems. Here we discuss sense organ regulatory genes, including sign oculus, brain 3, and eyes absent, that are expressed in Cnidarian sense organs. 2, they assess the sensory features of the planula, polyp, and medusa life history stages. And 3, they discuss the physiological molecular data that suggests sensory and neural processes in sponges. So these are all organisms that don't really have you know, complex or central nervous systems, but they do have these sort of neural signaling aspects, and they have sensory organs. So this is something that, you know, if you really want to know more about like sort of the origins of these things, Cnidaria is the model system you want to look at. And I mean, when I say model system, again, this is a whole host of model organisms that you can draw from, and you know, this is going to have you know, similarities to sense organs and kidneys and other things in bilaterians. So this kind of goes through the state of the art in Cnidaria. And then this paper is on a short history of nearly every sense, the evolutionary history of vertebrate sensory cell types. So this is a nice paper that goes through all of the different types of major sensory cell types found in vertebrates, their phylogenetic distribution, and presents a scenario for the evolutionary history of various sensory cell types involved in several cell type diversification and fusion events. And so this is, again, it tells you something about all these different sensory cell types. And, you know, like, so for example, they go through a selection of sensory cells. The vertebrate sensory, this is a rod, this is a hair cell, an innervating neuron, this is a mechanosensory cell in insects, this is an external sense organ, and then this is a sheath cell on a hair, I think, this is the direction of the stimulus, so it's stimulating this hair and it's coming down into the sheath cell. So you can see like all these different, you know, relationships between well, in C they show the direction of the sensory stimulus as well. So you can see the connection now between sensory stimuli and these cells, and then understanding what the output of those cells are, if it's continuous or discrete. And that's something we can talk about at another time. And then finally, this paper on the development and evolutionary sensory cells and organs. This is an editorial in developmental biology, and this kind of goes through some nice papers. Detlev Remp, who's an author who's done a number of papers on this, and they kind of go through some of this. So they talk about some of the new kinds of data that we're collecting on the evolutionary history of sensory cells and organs. New conceptual approaches are urgently needed to allow us to frame testable hypotheses for the evolution of sensory cells and organs. I think we could also use computational methods for it too, just to see like what, you know, the diversity of these kind of cells yields in terms of behaviors or outputs, and then looking at maybe at different hypothetical outputs or different hypothetical cell types. You know, what's going on at the molecular level, what's going at the cellular level, what are the stimuli, what does it look like when you stimulate these cells at different degrees of stimulation, and then what are their outputs look like. And those are what could all be done in silico quite well, I think. So this is from a special issue, Development and Evolutionary Sensory Cells and Organs. This was from a workshop in 2016. So this kind of goes through some of the papers. Todd Oakley contributed something here that highlights the duplication and divergence of genes, cell types, and organs, but these only form part of the picture, and there is an importance of rearrangements and co-options of pre-existing components. So in complex nervous systems, things often get rearranged and co-opted for different purposes. So this is kind of like exaptation. You have something that's used for one thing, you co-opt it for another thing. So that's all I'm going to talk about today. I hope that you found that useful. Thank you.